Good day, everyone. My name is Samir Ahmed, and I am an undergraduate student at SUNY Westbury. And the topic of my research is sexual dimorphism in microglial behavior and its effect on glioblastoma multiforme. So glioblastoma multiforme is one of the most common and aggressive brain tumors, and GBM patients have a very low survival rate of only 6.8%, and the average length of survival being only 12 to 18 months. Recent studies have also shown that males are 1.5 to 3.5 times more likely to be afflicted with GBM than females. And when diagnosed, they're also found to have a lower survivability rate. So when discussing GBM, microglia are especially relevant since they comprise of over 30% of cells in glioma biopsies. And pro-inflammatory microglia are anti-tumorigenic, whereas anti-inflammatory microglia are pro-tumorigenic. Interestingly enough, females contain a greater number of inflammatory microglia than males. And so the question arises, could the disparity in GBM outcomes be explained by the sexual dimorphism we find in microglial function? So in order for us to explain these different disparities that we find in GBM, we needed to observe the differences between female-derived microglia and male-derived microglia. One of the questions we asked was whether female-derived microglia were more inflammatory than male-derived microglia. We also proposed that the sex-linked steroid hormone estrogen, which is more prevalent in females, played a role in promoting microglia towards a pro-inflammatory shift, as well as directly affecting glioma growth and migration. So in the following experimental design, we took our N9 male-derived microglia and BB2 female-derived microglia, and we put them in the following conditions: untreated or treated with LPS, which is a lipopolysaccharide, which promotes a pro-inflammatory shift in the microglial cell lines, or we treated them with IL-4, which is a cytokine which promotes an anti-inflammatory shift in the microglial cell lines. The cell samples were collected at 4 and 24 hours post-treatment. And so over here is a picture that shows the morphology of the N9 and BB2 microglial cells 24 hours after the indicated treatments. Ramified shapes indicate resting microglia or M2 morphology, while amoeboid shapes indicate activated M1 microglia. Interestingly enough, a comparison of the LPS treatments between the N9 and the BB2 cell lines, as you can see over here in the picture, the comparison shows that the BB2 cells had a more dramatic morphology change to an amoeboid shift when treated with LPS as compared to N9, which did not have such a profound shift when treated with LPS. So this shows that BB2s had a stronger M1 or pro-inflammatory shift than the N9 cells when promoted by the LPS. So RNA was also harvested from both the N9 and BB2 cells to assess the differences in the levels of TNF-alpha, INOS, and IL-6, which are pro-inflammatory cytokines that are associated with M1 microglia. And we find that in the comparison between the untreated N9 and BB2 cells, the levels of TNF-alpha and INOS are higher in the BB2 cells, as you can see in the brighter bands over here. And in the picture, as you can see to the right, we also find that the same pattern for the rest of the treatments, and IL-6 levels for the most part seem similar in all treatments. between the two cells, except for the 24-hour treatment of IL-4, which is something that we will investigate in our later projects. So another question that we had asked was whether estrogen polarized microglia to an inflammatory phenotype. So in the following experiment, N9 cells were placed into two categories, with estrogen and without estrogen. And these two groups were untreated or treated with LPS, or they were treated with IL-4. Samples were then collected at 24 hours post-treatment. And so the RNA was harvested from the N9 cells, and we also assessed the quantification of TNF alpha fold. So if there's a higher TNF alpha fold, this indicates that the cell lines are going towards a more pro-inflammatory phenotype shift. And interestingly enough, our results were quite surprising because we found that the TNF alpha fold change actually decreased when cells were exposed to estrogen, and they were then treated with LPS. So this indicates that estrogen actually had a suppressive effect on the microglial cells, reducing their production of the pro-inflammatory cytokine TNF alpha. Finally, our last question was to see whether estrogen had a direct effect on glioma migration and growth. And so we performed a scratch migration assay, and the GL261 glioblastoma cells were given identical treatments that the N9 cells were given in the previous experiment. So we were observing the extent of cell migration after 48 hours. So if you look at these controlled conditions, you can see that after 48 hours, the glioblastoma migration actually increases. However, when we look at the conditions where they are directly treated with estrogen, their migration had significantly decreased compared to the controls. And so overall, all of these results showed us that female-derived microglia are inherently more inflammatory than male-derived microglia. And the results with estrogen showed that the sex-linked steroid may not be responsible for this difference between males and females, since it actually promotes an anti-inflammatory shift in microglia. However, estrogen did have a direct effect on glioma migration, since it suppressed it when the gliomas were directly treated with it. And lastly, thank you everyone for joining me today and listening to my talk. I would like to thank my research mentor and advisor, Dr. Jillian Nissen, for her support and guidance, as well as my lab partners and colleagues, Mohammed Mian, Tanya Kumar, and Marianne Johnson. And I would also like to thank Dr. Sturka and Dr. Costa Martinez at Stony Brook University, who provided us with the N9, GL261, and the BB2 cell lines. Thank you again. And if there are any questions, please don't hesitate to ask.